In this video, we will discuss some of the prospective therapies against PDAC or pancreatic adenocarcinoma. So, some of these topics might coincide with our previous topic, but in this video, we will discuss something in depth compared to our previous topics. So, let us start with what are the cytokines that are released by these PDAC tumor cells. Some of the cytokines that are released are TGF beta, CXCL5, GM, CSF. So what it does is it will mobilize this bone marrow to produce these immunosuppressive MDSC or T regulatory cells, and that will inhibit the function of the CD8 T cells. And also these T regulatory cells will block this tumor activated M1 phenotype tumor associated macrophages, and simultaneously this because of the metabolic changes in the tumor microenvironment of this pancreatic adenocarcinoma will secrete higher amount of this lactate that will also inhibit this CD8 plus T cells function by reducing the pH near the tumor, or we can say in the tumor microenvironment. So these are the cases. These are some of the conditions by which this immunosuppression around this tumor cells of the PDAC will occurs, and some of the other ways by which this immune suppression occurs is by the interaction of these cancer cells and the PSC cells. So what happens is in this case is that this PSC cells production, because PSC cells interaction with cancer cells will lead to the production of this extracellular matrix that will act as a shield or prevent the dendritic cells to come in contact with this tumor antigen. And simultaneously these cancer cells also release this IL-6 cancer cells and MDSC cells, and this will also promotes this formation of this extracellular matrix. And so what are the strategies that are being targeted right now? So one of the strategies, popular immunotherapy strategies, is the immune checkpoint blockage immune checkpoint therapies. So in this case also the one of the blockade immune checkpoint therapy that has been targeted on this PD-L1 receptor. So in this case, if these PD-L1 receptors get blocked by this antibody, then the cancer cells function to inhibit this the T regulatory cells will be stopped, and the microenvironment near the tumor cells will be less immunosuppressive. At the same time, other therapies that are being targeted is this CAR T cell therapy with a receptor against this mesothelin, which is highly expressed in this PDAC pancreatic cancer cells. So in this case also the CAR T cells will be able to distinguish themselves from PDAC cells and the healthy cells, and can target or act against these cancer cells. So these are the two strategies that are that have shown some positive results. However, the results are not to a level that it can be commercialized. So there are still research needs to be carried out so that these therapies might become more efficient and can be used commercially. So this is some of the detailed explanation, is that this mesothelin is highly expressed in this PDAC tumor cells, so that its antigen can be detected by these dendritic cells, and these dendritic cells can then activate the CD8 T cells, and CD8 T cells will then act against the mesothelin associated antigen, and that are usually present on the PDAC tumor cells. And this as I have already said, that is PDA1 is one of the immune checkpoint blockade that cancer cells use to suppress the T cell response by blocking or by deactivating the T cells through the interaction of this PDL1 and PDI on the T regulatory cells. So this is one of the prospective strategies that are under study against the PDAC or pancreatic adenocarcinomas tumors. If you need to understand more about these PDAC prospective therapies, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.